In the second part of the lecture on biology, we will focus on the organism. In the first part, we have seen the molecules. Now we will see the organisms. So we start from here. If you remember, this was the last slide where essentially we compared the three types of organisms on Earth. Let's say that these are the viruses, the prokaryotes, and the eukaryotes. Now, viruses are not quite always considered living organs by most standards, because they are somehow, but they are generally accepted as such, but they have not really a nucleus and not really a membrane. They have no organelles. And that's the most important point. They cannot reproduce on its own. However, especially these days, of course, they are very popular, and we will see why. The superior to the viruses are prokaryotes. Prokaryotes, bacteria, essentially. We will see that bacteria is the most important prokaryote. They also have no nuclear membrane. They have a cell wall, though, that divides the cell from the liquids. In some cases, they have no organelles, apart from but in some cases, they have some. There is a capsule surrounding it, and there are three main types. Actually, generally, we talk now about two main types of prokaryotes. We will see in a second. And finally, eukaryotes, in their case, the nucleic acid, unlike viruses and prokaryotes, have his own membrane, so it's separated from the rest of the cell. The plants have a cell wall, all the plants, contains many organelles, we will see. The external membrane is a bilayer of lipids, and is specialized by thousands of different sizes and shapes. So we will, of course, we are eukaryotes, all the animals and plants are eukaryotes, but we will shortly have a look also to virus and bacteria before going to the superior cells. So let's start from viruses. Now, you see here, the properties of the virus is in the nucleic acid, in the shape, and in the strandness of the nucleic acid. So the nucleic acids can be DNA, RNA, or both DNA and RNA at different stages in the life cycle. The virus that you see here is a T4 virus. It's not the virus attacking human cells. It's not attacking eukaryotes in general. It's attacking bacteria. And is a DNA virus. The SARS-CoV-2, which is the virus, as all of you know, is responsible for COVID-19 disease, is an RNA virus, and is single-stranded, while this virus here is double-stranded. In some cases, viruses are double-stranded with regions of single-strandedness. The shape is very different. It can be linear, circular, or segmented like this one. Circular is typically the famous coronavirus, is circular. Here you see how essentially they work. This is a bacteriophage, so a virus of bacteria, and that's the way they work. They attach on the surface of the bacteria, and then they inject their virus in the bacteria. This is a cartoon, but down here you see an electron microscope image. Keep in mind that the size of this virus is only 20 or 30 nanometers. It's extremely small. And you see the injection, you clearly see the collar and the syringe, the legs of the virus, and the syringe where it is actually injecting the DNA inside the bacteria. The bacteria will be fooled, will treat the viral DNA as his own DNA, and then will use his machinery to build new viruses that then will make the virus explode, and then the viruses will go to attack other bacteria. This is exactly the same process that happens for viruses in the mammalian cells. So these are bacteriophage, but it's the same story for the viruses in mammalian cells. These are again phage T4. This picture is interesting. It goes to radiation. These viruses were exposed to radiation. And you see some of them have a black head. Some of them have a white head, you see it here. And in some of these white heads, you can see sometimes the DNA coming out of the head. So radiation was able to induce a hole in the capsid, and the DNA is going out from the hole of this capsid. Radiation is generally, we will see, useful tool for vaccine, because to make a vaccine, one very classical method is to use inactivated virus. So you can inactivate the virus through the radiation, like is shown here, and then you induce the immune response in the host and the vaccine. Now, of course, we cannot avoid to talk about the famous SARS-CoV-2, this coronavirus type 2, which is responsible for the acute acute respiratory syndrome. This is actually an electron microscope image with of course with false colors that show these spike proteins that you see. This is a famous design of the virus, which is often used. Actually, these spike proteins are really extremely important because they are those responsible for the infectivity of the virus. This is how the virus gets attached to the cells. The rest of the virus, as you see, is done. These are the spike proteins. This is the envelope with envelope proteins and membrane glycoproteins in the envelope. And this is the single-stranded RNA molecule of about 30 kilobase pairs that make up the genome of this virus. These are other very nice, of course, everybody's studying now these viruses, and these are other very nice pictures at the electron microscope, pseudo-colors, of course, but very spectacular, showing the virus. Here is shown in yellow, here is shown in pink, here is shown in orange, and here is shown in yellow, attacking human cells. The shape of the cells that they are attacking, in particularly here, you see clearly, but also here and here, is very strange. This is called blebbing of the cell. So the fact that the virus attacked the cell induced the apoptosis in the cell. So these are cells that are dying before releasing the viruses outside. And apoptosis is one cell death pathway that we will study when we will see the cell death in mammalian cells. So how the SARS-CoV-2 works and how it leads to pneumonia, because essentially the COVID-19 is a pneumonia, it's a form of pneumonitis. Well, essentially, the process is the following. The SARS-CoV-2 attacked the cells using a very common receptor that we have on the membrane of most of our cells. This is also why it's so effective. It can attack almost every cell. And this is a receptor called ACE2. Now, once the virus is internalized, because attacks there, then is internalized, what happens is that there is a genomic RNA release. So release the RNA from the envelope. At that point, the cell, thinking that this is a normal RNA, starts what I told you last time, starts the translation of the RNA into proteins. And these proteins are actually viral proteins. And then at the end, the new virus will be released by the cell and will go to infect other cells. What happens is that at this point, these numerous viruses around, they are activated through different mechanisms, and especially, this is the important part, the action of the virus lead to the release of cytokine and the accumulation of neutrophils and monocytes and macrophages. These are essential excuse me. These are essential for the inflammation process. So that's where they cause inflammation in the lung and then pneumonitis. Now, in this cartoon, you see the strategies that they are now under study for the vaccination for SARS-CoV-2. There are different strategies. Some of them are very innovative, those based on proteins or on virus-like particles, and those based on viral vector vaccines. The most traditional, conventional, and actually probably also the most successful strategies here are those based on weakened viruses or inactivated viruses. These are also the strategies generally used for vaccination for the flu or many other diseases. In this case, essentially what happens is that you have an inactive virus. It can be inactivated with radiation, can be inactivated very often these days with chemicals like formaldehyde, sometimes with heat. So essentially you go to high temperature. And then what happens that when you inject it in a host organism, sometimes this happens in the eggs. It is injected in eggs because you can have many eggs. These viruses are now unable to induce to replicate themselves, but the antigens are here. The antigen presenting cell are the dendritic cells. We will see they recognize and they will trigger an immune response. The immune response will give you antibodies, then you extract these antibodies from the egg, and this is your vaccine. This is why these days we are also studying the possibility of using not gamma rays or chemicals for viral inactivation, but to use heavy ions. This is apparently a strange idea, but if you remember how the heavy ion works in comparison to gamma rays, you can understand the logic. You see, in this cartoon, you see that if you want to inactivate the virus, you need to cut the RNA inside the virus. But if you do it with photons, you will need many photons, and these many photons eventually will damage also the spike proteins. So you will reduce the ability of the inactivated virus to elicit an immune response. If you do it with heavy ions instead, because if one heavy ion is enough to cut the RNA, then the damage to the surface will be very little. And these are, you see, Monte Carlo simulations showing now a realistic shape of the coronavirus, and the tracks of gamma rays are, of course, they are electrons generated by the gamma rays, and these are this is a track of heavy ions going through the coronavirus. Okay. From virus, we move on. The next step are the prokaryotes. The prokaryote, actually, at the moment, it is believed that all the organisms come from a universal ancestor. And from this universal ancestor, it was split essentially in three domains. The domain eukaryotes, the domain archaea, that previously were considered a form of bacteria, and the domain bacteria themselves, that are also divided in proteobacteria, chlamydias, spirochetes, cyanobacteria, and gram-positive bacteria. You probably know already some of these names because there are many diseases and many antibiotics. Antibiotics are the drugs against the bacteria that are responsible for all cause of disease in humans. Of course, unlike for virus, for bacteria can be specifically killed by antibiotics that are specifically toxic for bacteria. So the treatment of bacteria disease is easier than the treatment for viral disease. Bacteria, you see here, bacteria in the bloodstream, these are actually macrophage, again, cells of the immune system that are actually attacking bacteria and destroying bacteria. You see, bacteria are, of course, much smaller than than the eukaryotic cells. Eukaryotic cells are approximately 10 microns in diameter. Bacteria is approximately 1 micron in size. And you see, this is again the same picture I showed you before, but this time you see in green, you see the virus, and in blue, you see the bacteria that are attacked by the virus. The shape of the bacteria is amazingly different. You see here a number of very well-known bacteria. You see that this is the Streptococcus pneumonia. This is the bacteria that cause pneumonia, not the viral pneumonia, but the bacterial pneumonia. And there are many others very famous. The Helicobacter pylori, for example, is very well known now to be involved in gastritis, in all the inflammation of the stomach are related to Helicobacter pylori and increase the risk of gastric cancer. Other very well known here are, for example, this is the Vibrio cholerae, which is responsible for cholera. This is the Salmonella, which is responsible for Salmonella, and so on and so forth. The shape, you know, is pretty much like that. Externally, some bacteria, not all of them, but some bacteria have this flagella, they are called flagellum, you see, or pilus. So they have some kind of external hairs. There is a capsule and there is a cell wall, so they are quite thick in the membrane. Then there is also a plasma membrane. This is it's called the nucleoid. In reality, the DNA inside the bacteria, there is no membrane for the DNA, so it's dispersed in the cytoplasm, and the cytoplasm contains some organelles, especially ribosomes. Ribosomes, of course, are essential for producing protein, so if the bacteria has to produce its own proteins, he needs ribosomes. Now, if we think to the human body now, so we shift just a second, but I'll show you why. In our body, there are approximately 10 to the 14 cells. However, approximately 50% of these cells are bacteria. So, which can be surprising if you think about, you think that you are a eukaryote, and our cells are eukaryotic cells, but 50% of our cells are prokaryotes. So in our organism, they are both eukaryotic and prokaryotic cells. So approximately 5 per 10 to the 12, 10% of the prokaryotic cells make the solid tissues. The others are the circulatory and lymphatic systems. We have about 3 per 10 to the 13 erythrocytes, the red cells in the blood, are very, very numerous. The diploid genome, this we have seen already, is about 6 per 10 to the 9 base pairs. With the population of 6 per 10 to the 9 individuals in 2013, that I should revise because now it's more than 7 per 10 to the 9 individuals, the total length of the human DNA is approximately 10 to the 20 kilometers, which is in megaparsecs, so is more than the Milky Way, than the diameter of the Milky Way. If we put together all the DNA of the humans, this will be a string that will be longer than the Milky Way. But as I said, half of our cells are bacteria. Where are these bacteria? Most of these bacteria are in the intestine. These are intestinal microbia. And they are involved in many functions. They are involved mostly in digestion, mainly through the fermentation of carbohydrates, but they also have several other functions, in particular in the immunity. The disruption of the gut flora is detrimental to health, and which is a relatively new discovery. You see, this is the cover of Science in 2012, when the word, the second human genome was created. The first human genome, of course, is the DNA. But recently, in the past few years, it was clear that we can define another genome, another fingerprint of our own body, and this is the population of bacteria in our intestine. This is really a second genome in the sense that is very individual, and even within the same individual can change with time quite radically, depending on diet, metabolism, and other factors. the metagenomic basis of therapeutics is now merging pharmaceuticals to environmental factors in the body. This is called metagenomic basis. Essentially, bacteria becomes now a target also for therapy of different disease. Okay, with this, we stop with the prokaryotes and we move to the next step, which is now the eukaryotes. Now, the eukaryotes, as I said, have a nucleus with the DNA, a cytoplasm, and a membrane. And we have seen already these three sectors separately. But which is at the most important, they have a life, and the life is called the cell cycle. And the cell cycle, you see, is divided in different parts. The so-called G0 phase is not shown here, is a phase where the cells are essentially resting, they are not moving. Then you have the so-called G1 phase, gap 1, followed by the S phase. The S phase, S stands for synthesis, so this is the phase where the DNA is duplicated. So you double the DNA. In the G2 phase is another gap phase, the second gap phase that precedes the mitosis. In the mitosis, one cell becomes two cells. You see here, this is the mass of the cell as a function of the time, and these are the different phases. You see, these are proteins, RNA, and DNA. In the G1 phase, you have an increase in the number of proteins, an increase in the transcriptome, but the DNA is one copy, or to better say, is always, if you want, we can say two, because it's actually one chromosome from the father and one chromosome from the mother. But during the S phase, every DNA strand will be duplicated. So now you have like 4 strands. Each chromosome has 2 double strands. And this is also in G2. And you see the increase in the number of proteins and the transcriptomes also increase along the cell cycle. So the DNA in mitosis and in G1 have different shape. You see, in mitosis, everybody knows the DNA assume a compact form, very visible at the microscope, because this now the width here is about 0.2, 0.3 microns, and this is, you see, 10 microns. So it's really, they are really big, clearly visible in an optical chromosome. In fact, they are known since many times. But actually, the chromosomes are also present in interphase. This is chicken, this is human, but you see, also in G1 there are chromosomes. So the DNA is not one single strand of 2 meters. It's actually divided, it's chopped in a number of different chromosomes. In the human body, we will see now, there are 46 chromosomes, but this number is not the same in different species. Together, you see here, together with the DNA, there are always proteins, and these proteins are called chromatins. Why is that? Because I told you last time, in any case, you have a 2-meter string, and you have now to pack your 2-meter string in a 10-micron nucleus. How do you do that? You do that wrapping the DNA along the proteins. The first proteins, you see, these histones, these are called histones, make a structure which is called beads on a string. Essentially, you wrap on one disk, and then you connect one disk to another disk. At this point, you can wrap all the disks, forming the so-called 30 nanometer thick fiber of packed nucleosomes. Then you go on in packing, you know, and you do this section, and finally, when you go into mitosis, this becomes much bigger, much dense, giving rise to the so-called mitotic chromosome. So you see, the results is that each DNA molecule has been packed into mitotic chromosomes that is 10 1,000-fold shorter than its extended length. Moreover, how compact is the chromatin? I mean, the fusion of the DNA with the proteins in the nucleus is called chromatin. Not necessarily compact in the same way. Some areas are very compact. They are highly methylated. You remember these epigenetic systems of methylated, and it's called heterochromatin. Another region instead is highly decondensed, is not much methylated, is transcriptionally competent, is accessible to nuclease, while heterochromatin is not. Essentially, heterochromatin is where the cell is putting the transcriptionally silent genes. Euchromatin is where the cells wants to put the genes that are frequently transcribed. And this is, of course, because then it's much easier the access in an area where it's not compacted in chromatin. This can actually be seen also in the chromosomes. You see, in the chromosomes with different staining techniques. For example, here you see in orange the heterochromatin. Telomeres are the capping of the chromosomes. We will come back to them. And in blue is the euchromatic regions. Now in this video you see the wrapping of the DNA. So how can we really compact?
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated.
The same is not exactly true that they're not always present. In the condensed form, they are not always present, but in the more decondensed form, they are present. And this is the mitosis. You see, now the DNA which has been duplicated in S phase flow into the cells. This is now the DNA replication. So how
using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells.
But this we will see later. So for now, let me go back to the epigenetics and show you. You see that the epigenetic mechanisms that we mentioned already, like methylation, the importance of this epigenetic mechanism in regulating the different transcription of the cells. You see that with methylation you make more compact, and you make the DNA inaccessible and the genes inactive. And of course, somehow we will see methylation is related to aging, but it's also relating to cancer, because of course, if you want to bring the cells back to their original embryo stem cell state, you have to demethylate and to make an histone modification that makes the DNA available. So you understand that the epigenetic mechanisms are becoming more and more important in therapy tools, not only in basic understanding of the biology. In addition, you see now we start to see here a little bit more about the shape of the chromosomes. as I told you, we will come back to this one, but the karyotype, this is a karyotype, so it shows the chromosomes that have been, of course, pictures of the chromosomes have been coupled. There are 46, oh, I'm sorry. There are 46 chromosomes and 2 X chromosomes. Of course, the 2 chromosomes come from the 2 parents, one from the father and one from the mother. Was one pair was in the sperm and one pair was in the egg. These germ cells, so the egg in the woman and the sperm cells in the man, are the only cells in the human body not having 46 chromosomes, but of course they must have 23 chromosomes, and exactly they must have 22 autosomes plus 1 sex chromosome. The sex chromosomes can be X or Y. For the egg will be always X, but for the sperm it can be either X or Y, so that if the fusion gives an XX will be a female, if the fusion gives an XY will be a male. The chromosomes are also divided in gene-rich and gene-poor. Some chromosomes are very gene-rich and other chromosomes are very gene-poor. What is interesting is that the gene-rich chromosomes are more in the center of the chromosome, while the gene-poor chromosomes are more in the periphery. So it's a sort of the nucleus, I'm sorry, I meant the center of the nucleus. It's a sort of protection of those chromosomes that are gene-rich. This is now the second part of the movie that shows you the S phase.
We are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
So just a note, as you see, one strand is copied very easily and continuously. The other strand instead has to be copied backward, and that's because of the story of the 3', 5'. If you remember, you can only go in one direction. You can go 3', 5'. So if you go 3', if you open up, one strand is 3', 5', but the other strand is 5', 3'. If the enzyme only go 3', 5', you have to, for one strand, you can go fast in one direction. For the other one, you have to go backward. But at the end, you will have the duplication of the total DNA. And finally, the mitosis, you can see here in these movies, I mean, this is a spectacular example of synchronized.
New cells are formed by cell division, in which one cell splits to become 2 cells. Cell division involves 2 processes. In the first, called nuclear division, the nucleus divides. In the second process, cytokinesis, the cytoplasm divides, producing 2 cells.
Now, so we have seen the different phases, okay? G1S, G2M. But how is this process regulated? Well, you see it here. Of course, the regulation of the process, the switch from one phase to another, is generally regulated by, of course, eventually it's regulated by the DNA. But you know, the DNA change some proteins that, not by chance, are generally called cyclins. You see here, this is cyclin E, cyclin A, cyclin D is this one, and the cyclin B is this one. You see that from G1 you start to produce a lot of cyclin D, But the cyclin E first increase, then starts to decrease with the increase of cyclin A. Cyclin A increase in S phase, then in G2 decrease, because the cyclin B is the last one that brings the cells into mitosis. This is also shown here. You see that the increase of the cyclin that bring into mitosis. And what is interesting, you can see here, what is interesting is that you can actually induce mitosis into cells that should not go into mitosis. If you do a trick, let's say, because the mitosis is triggered by the MPF, by the mitotic promoting factor form, you see, which is, you see, this is cyclin B. This is this is a chemical which is used to actually bring in this level, you see, to have the production of MPF using a chemical in a phase which is not mitotic. When you do that, something very strange can happen. You see, in this picture, there are three cells. One cell is here, one cell is here, and one cell is here. These three cells are in three different phases of the cell cycle. This one is in G1, this one is in G2, and this one is in S. How do I know that? Because the condensation of the chromosomes was induced in these cells with this chemical that as I told you, release the cyclins, so fools the cells, and bring to the condensation of the chromosomes. I can tell that this is a G1 cells because there are now 46 chromosomes, but each chromosome is only one strand, one DNA strand. So it's before S phase, while this is a G2 phase because now each chromosome, you see here, has two strands of the DNA. So they are ready to go into mitosis, if you want, into the real mitosis. Into the real mitosis, you need to do the two strands because then one strand will go in one daughter cells and the other strand will go in the second daughter cell. This is an S phase cell because in that case, part of the DNA is duplicated and part of the DNA is not duplicated during the S phase. And this brings to a strange configuration here, you see, where the condensation and decondensation is sparse. There is this DNA which is left in between without proteins. So with this technique known as premature chromosome condensation, you can actually visualize the cells in the three different phases of the cell cycle. This is a real picture under the microscope. I did it myself, so I'm very proud of it. And only by luck, I found three cells in three different phases exactly in the same field of the microscope. This is how the human karyotype looks like for a male, because you see there is an X and there is a Y. You see that the chromosomes, these are 22 autosomes and the two sex chromosomes, have different size. I mean, the numbers 1, 2, 3 up to 22 generally correspond to a different size. So you go from the big chromosomes like 1 and 2 to the very small chromosomes like 21 and 22. X is, of course, an exception. These are the sex chromosomes. And you see that some chromosomes are metacentric. So the centromere, which is the area where the DNA strands are kept together, the two DNA strands, you know, from after the S phase are kept together, can be in the center of the chromosome, can be submetacentric, or can be even acrocentric, which means that the two strands are actually kept at the end of the chromosome. And that's how it looks like. This is a cartoon, of course, but this is a real karyotype. Instead, colored with the technique known as M-fish. We will describe this technique. These are actually my own chromosomes. You see, it's a man, X and Y. The different colors divide the different chromosomes. They look the same, but actually one is maternal and one is paternal. And you see that you have 22 autosomes and 2 X chromosomes, 22 pair of autosomes, of course, and 2 X chromosomes. And this is a picture taken from cells in M phase, in mitosis. In fact, you see very clearly that each chromosome is composed of 2 chromatids, and chromatids are called, so 2 DNA strands, so after the S phase, and these 2 DNA strands created in the S phase are kept together at the level of the centromere. We will then talk about the structure of the capping of the telomeres in the next part of this lecture.